Okay, this is the last section for Unit 2 in Pre-AP Biology. We'll call this one 024, and it's going to talk about enzymes, which are a special group of proteins which actually allow us to speed up processes that go in our bodies naturally, doing them faster and also with less energy. Some of these reactions would take energies more than 37 degrees Celsius or 98.6 degrees Fahrenheit, which would be bad for us, or even lower temperatures than that 37 degrees Celsius, which would be bad with us. But enzymes are any kind of a special protein group that acts as a catalyst to speed up reactions. They do this by lowering the amount of energy required, which is called the activation energy. And I'd like to go ahead and show you a little animated view of this right now.
Imagine a chemical reaction with the general form AB yields A plus B. In order for this reaction to occur, the reactant molecule AB must acquire enough energy to disrupt its stable electron configuration so that its electrons can rearrange to form A and B.
Okay, so this reaction took place, and what has to happen is we have to put in a lot of energy, and the activation energy rises all the way up to there, and that's probably too high for our bodies. What we want to do is we want to break this. Again, that process is called hydrolysis. We want to break this from the AB molecule into an A and a B and come up with these products. What does an enzyme do?
This energy barrier is called the activation energy of the reaction. In living cells, enzymes serve as biological catalysts, speeding up reactions. Enzymes accomplish this task by lowering the activation energy of the reaction, making it easier.
Okay, what I want to show you on this one is notice how often we get this AB configuration and how often we get the old version. And you see the process happens much, much, much faster with the help of an enzyme. This is actually showing the enzyme on the substrate, and it shows you the process of hydrolysis and reducing the processes all the way through. This is showing you a competitive inhibitor. And basically what's going on is this is the normal process, and if we go into
Non-competitive
this,
inhibitors bind to a location on the enzyme other than the active site. For
and you see it actually going into that site, it changes the shape of this active site, and what it does, it makes it less efficient at doing what it's doing. Every once in a while, one actually sticks and does work, but in a normal enzymatic shape, all of them work. And notice that it is reusable. It can go back and forth and back and forth and back and forth. Okay, and we are done with that particular one. So there's the enzyme. There's the substrate. This is called the active site. And the active site actually is like a lock and key. Only one substrate fits into one very specific enzyme. It can only work with one particular type. And then the enzyme substrate actually comes into this location where the substrate's actually attached to the enzyme. And then because of the close association, we can get that hydrolysis to happen and we can get the products. And the enzyme is, again, reusable. Let me go ahead and show you this shockwave. And this is going to be the substrate. There's the enzyme. And notice it's a whole lot of amino acids put together in a very specific shape, and it's not quite as round as some artistic views of it. This is a more realistic enzymatic shape.
In living organisms, most chemical reactions are catalyzed by proteins called enzymes. The three-dimensional structure of an enzyme is crucial to its function. If this structure is disrupted by exposure to high temperatures, for example, the enzyme stops working. The enzyme binds reactants in its active site, which is specially shaped to fit the reactants. The reactants on which enzymes act are called substrates. In this
And notice it's a very specific enzymatic shape for that one particular substrate.
example, glucose is the substrate of the enzyme hexokinase.
Glucose, OSE, which is actually showing you that it's a sugar, and kinase, which is actually showing you that it's a cutting version, hydrolysis.
The enzyme and substrates are represented here in an artist's drawing. The substrates include the atom A and the molecule BC. The substrates enter the active site of the enzyme. By holding these atoms close together, the enzyme reduces the amount of energy the atoms need to enter the transition state. In the transition state, the bond between atoms B and C is broken,
Hydrolysis,
and a new bond between A and
dehydration
B is
synthesis.
created. When the new bonds form, the products are released, and the enzyme molecule is ready to catalyze another reaction. Each enzyme is specific for only one reaction. For example, this enzyme could not catalyze a reaction between atom A and molecule BD.
Again, it's a lock and key, so very specific to very specific enzymes or substrates.
The fit between an enzyme and its substrate is described by the lock and key model. The enzyme is the lock, and the substrate is the key. Only a specific substrate will fit the enzyme's active site. Another model of enzyme-substrate interaction is called the Induced Fit Model. It proposes that once the enzyme binds the substrate to the active site, the enzyme changes shape slightly to bind the substrate even more firmly. This places a strain on the existing bonds in the substrate, thereby lowering the activation energy needed to break the old bonds and generate new ones.
Yeah, the big thing is that it's very specific and it does lower the activation energy. This is a site that's going to hopefully allow me to watch the super blood lunar eclipse that's going to happen tonight that I told you guys about last week. It is supposed to happen, well, around 10 o'clock p.m. tonight is going to be the best view. Just hope the skies are going to be as clear as they are now. Okay, so what affects enzymes? Enzymes are proteins. Proteins hate changes in temperature. Too hot, too cold, it changes the shape of the protein, and the protein doesn't do its job. It's very specific enzymes. That means the enzymatic reaction doesn't work. So enzymes have a very optimal temperature. Notice that the typical human enzyme happens at around 37 degrees Celsius, which is the body temperature of humans. And notice that the rate actually tails off if it gets too cold and tails off even faster if it gets too hot. The optimum is 37 degrees. If you're a thermophilic or you're a type of living form that actually likes heat, your enzymes happen at much hotter temperatures. So notice that if we raise our temperature to that point, the enzymes aren't working at all. And if we cool these bacteria down to our temperature, they don't work very well. So these bacteria usually are not going to affect our bodies because they can't survive. pH, or the power of hydrogen, talking about how acidic it is or how basic it is. Enzymes have a very optimal pH as well. Pepsin, which is produced in the stomach that actually starts breaking the peptide bonds in proteins, it has a pH of about 2, incredibly acidic. And then we have trypsin, which is actually produced in your pancreas. This also further breaks down peptide bonding. It actually says lysis and lysis, cutting down or cutting the proteins into smaller pieces. So when you ingest proteins from the food that you eat, we use pepsin to start breaking down the process. Actually, saliva's got some of this in there too. Break down the process, and trypsin continues the process. Outside optimal environments, enzymes denature, they break down, and they don't work. So we need to keep 37 degrees, 98.6 degree body temperature in order for our enzymes to work, and we need very specific pHs in parts of our body to get these enzymes to work as well. And that is it. Appreciate you stopping by. Thank you.